Hey! Okay, so we're back again for lesson 3 of my group problems. Um, so to start off, the f- there's 5 questions with various sub questions. So to start off, we're going over epigenetic inheritance. And so first question talks about um, a human disease called Angelman syndrome, which displays genomic imprinting. And so, supposing that that a recessive A is Angelman syndrome, while a capital A is the non-diseased allele, they asked what type of genotypes and phenotypes of the progeny if both the male and the female were heterozygous. And so, to begin off, I have my papers once again. So, question 1. This is the Angelman syndrome question. Hopefully you can see it. So all that I did is I took the crosses here. I crossed the parents together, and for the ones that would be displayed and would not be displayed over here, I kind of did everything in like a little dot pattern if they didn't express, versus the ones that are solid and do express. And so there'd be a 1-to-1 ratio in the sense of that there would be half of the party is non, and wait, let me make sure I'm getting this right. The pause, the capitals awkward. So the capital A's are the ones that are non, while the recessive A's are the Angelman syndrome. And so here's the genotypes listed out here with how much of a probability there would be. And then we have the phenotypes and what they would be. So part B, I'm going to send to this side because it's slightly easier. Not really, I lied. So question B is asking about about part A again. And they're asking about if you had a male that was heterozygous and a female that was homozygous recessive for Angelman syndrome, what are the genotypes and phenotypes of that progeny? And so, once again, part 1, part B. So I wrote out the parents again. So here they are. Here they are listed out. I did my little dotted line. And so the ones that would be expressed are all the little A's. That they would all be Angelman syndrome. So, make sure I did that. Oh, pardon me. What? Okay, what I meant to say is that they all have Angelman syndrome. I will have to look into that. But, so what happened is that these are supposed to be, if I listen to myself, and these up here, they're all Angelman syndrome because, well, actually, it depends on if it came from the father or not. And so that's probably where my brain's getting confused at this moment. So if it came from the father, they are there, but if they didn't, then yes. So that's what's happening. Anyway, moving on. Awkward. The part C is asking about a different question now concerning epigenetic inheritance, saying that a woman has triplo X syndrome, meaning that has she has 3 X chromosomes. However, forgive me, but I'm going to call it the XX, the XICS, which have been deleted from 2 of the X chromosomes in each of her cells. How many bar bodies are found in each of these of this woman's cells? And so I just kind of drew things out so I could see them. And so she has 3 X chromosomes. 2 of them have this deleted. So what I think would be is that there'd be 1 bar body, because this one's already active right here. So it needs another one to kind of crumple, I guess you could say. Um, I'm not quite sure, but so the cell only has one XIC, so one of the deleted ones need to continue to be active while the last isn't needed and compacted into a bar body. Oh, there we go. So that's much more coherent. I just need to read my paper. So moving on. One bar body because the other one still needs to be active. So part D is once again asking about bar bodies, but it's in a female cat. So the question says, suppose you isolate a cell from a female cat with 2 X chromosomes. However, the T6 gene on the X chromosome are non-functional. On each X chromosome are non-functional, while the XIST, just going to spell it out, genes on each X chromosome are functional. How many bar bodies are expected to be found in a cell? So once again, I drew it out just for convenience. So I have that this is non-functional and that these are functional. And so I will read what I wrote. So with the T6 being non-functional means both chromosomes would be active. But the XIC, or the XIST, however you say it, I'm just going to go with the flow. Since it's functional, causes one to go inactive. So I think there would once again be one bar body because there needs to be one. So that is that. So that's question 1. Question 2 is a lot of fill in the blank. So what I did is I'll read the question, but I'll have all the answers displayed that I wrote. And hopefully it makes sense. I'll just read blank. You can read what it says. And hopefully we all are happy. I don't know. Anyway, so here we go. So let me get these all displayed for you. So question A right here is asking blank is an example of a holoandric gene. And so the SRI, anyway, it's what helps develop male, with male development, making proteins for that. Part number B, right there. Merry Christmas. Blank is an example of a gene found on both the X and Y chromosomes. And so for this one, I put the MIC2. So it produces a cell's surface protein, and I put those there because it helps me remember. So part C, or there we are, kind of has like a little cross to do. So it's asking a cross is made between an IGF2 minus male, so heterozygous male mouse, and a heterozygous female mouse. What are the phenotypes of the offspring? And so what I did is I did a little cross. Now I did simplify how I went about this because I didn't want to write IGF a bajillion times. So I just wrote down the 2's because that's where the minus is. So in this case, the minus all being on this side of the cross, you get half and half for the progeny, half being dwarf and half being normal with their phenotypes. So on to part D. So in the blank pattern of inheritance, an allele can be dominant in one sex, while the same allele is recessive in the other sex. So it is the influence pattern of inheritance. Influenced. I think that makes sense. So part E, right there. Okay. So it says blank is an epigenetic process that occurs during gamete formation, while blank is an epigenetic process that occurs during embryogenesis. And so the first half is genomic imprinting. And I wrote down, let me get to my notes really fast. It's the first one because it allows for X-linked genes to have have a similar amount between the X chromosomes and in the female and the X and Y chromosome, or the X chromosome in the male. So that's why it's the first one. It also occurs during the gamete. Sorry, I read the wrong one. What I meant to say was that genomic imprinting occurs in just one gene, with one gene or an entire chromosome with a change somewhere in there. That's during gamete production. Sorry, I think I wrote down my word wrong, so I have to go back up here and yeah, formation. That's what I meant to write or say. Whereas the dosage compensation is what allows for X-linked genes to have a similar amount between the X chromosomes in the female and the male. And that's during the embryonic development. Okay, so part F right here. The question is, one of the topics discussed in lesson 2 involved the formation of a double-stranded RNA molecule. What two molecules form the two halves of this double-stranded RNA? And so in the notes for the lesson, the T6 and the XIST are the ones that are involved with that RNA. One doing one half, the other doing the other half during different processes. So part G, right down here, there we go. The question is, when reciprocal crosses give identical results, the alleles that control the trait in question are located on blank. When reciprocal crosses give different results, the allele that controls the trait in question are located on blank. So I have the autosomes are what control the trait in question if it gives an identical cross. Pardon me. Whereas the sex chromosomes are what happen if you get different results. And I think that comes down just to the fact, like, if it's X-linked, you have X-linked, for example, is the father only has one X anyway. So thus why it would be something slightly different. But then it also depends on what else is going on. So moving on to question 3. So question 3 has 4 parts to it. So hopefully this makes sense. Oh, I did write this down. Sorry. I was checking my notes. I couldn't see what I wrote, so I realized where I put it. So the answer are in the black boxes, and then I have the work displayed right here. So part A, let me just read it all for you. A woman's father has ornithine transcarbamylase deficiency. So they call it OTD. For my sake, I will use OTD. It's an X-linked recessive disorder producing mental deterioration if not properly treated. The woman's mother is homozygous and is unaffected. So it's saying that the mother is homozygous and the father has this disease, which is X-linked recessive. So, as the screen has moved, well, I've moved. You have the X chromosome on the father, which has the recessive allele. And the mother has 2 of the same chromosome, 2 dominant genes, because she is unaffected. So you do the cross, and you get that. I forgot what I was trying to find out. Oh, we're trying to find out the genotype of the woman and the woman's father and the mother. Genotype, right? So I just wrote them out because this is what makes sense to me, is that the father can only have that, where she's homozygous, so she has to have 2 of the same that are both capital. So I guess I just did this for fun. So moving on, part B. They're asking now about the woman who, if the woman has children with a man who does not have OTD, what is the chance that their first child will be a son with OTD? So I have their little blah blah blah. Can't think of the word. Anyway, here's their makeup, and here is what the cross that I did. So I came across the son would be half the chance because there's you have females or you have males, and then to get a son with OTD, he's the only one right there in the bottom corner. So there'd be a 1/8 chance that he would have OTD as their offspring. So part C. So here the question is, Identify the genotype of a male with whom the woman could produce a daughter with OTD. So the same thing, but this time we just don't know the male. So I went about trying to find a cross. So I put the female, and then I X and Y, and then figured out that, well, if I want a daughter, I just need to have two little t's together. And so I set everything else up and got that. So he needs to have a big, an X with a recessive allele to have a daughter with OTD as her as her. Well, I guess the question's asking to identify the male. Oh, bam. So then part D is consider the cross described in part C of this question. So talking about the daughter. What are the expected genotypes and phenotypes of this offspring? So just to show. So part C. So from this thing, I more or less just wrote out everything that had happened up above. So I have all the genotypes right here. And here's all the phenotypes that would be observed in this cross. So you have your females that would both be heterozygous and homozygous recessive, while also having a male that's dominant for that and a male that's recessive. And so that's part 3. So on to part 4, or question 4. So this one has 4 parts to it also. And it's talking about males and females with beards. And how, I'll just read the question. The presence of a beard in some species, we're talking about goats. Species goats is determined by an autosomal gene that is dominant in males and recessive in females. So question A is asking about the genotype and phenotypes of the male and female, or of the parents, if the cross produced 3 bearded and 1 bearded in a ratio amongst the male offspring. So this is what I have. So for my sake, I wrote everything out on top just to remind myself that males, a big B is dominant, whereas if it's a big B in females, it's recessive. So going about this, and luckily I'm just doing it about males for right now. So I wrote down that, we want 3 that have a beard and 1 without a beard. So I wrote out, males, they get big Bs that cause the beards because that's dominant with them. And so in this cross, to get this, you'd have to have a parent, the mother be, sorry, I have to read this, even though you've already read it. The genotype of the parents are heterozygous, so big B, little b. And the phenotype of the parents are no beard for the mother and beard for the father. Yeah, this is classy. All right, so then the next one's asking the same question, but in regards to females having a 1 ratio of a beard to a 3 ratio of no beard. So once again, I scribbled out what needed to happen, and then I was able to figure out that, the genotype of the parents would have to be the same because they flip-flop. So all these little b's are what give no beard, whereas the big B is what gives a beard because it's recessive. There we go. So that's why this ratio works with this, even though it may not seem like it. It's just backwards. It's just the opposite. They're like reciprocals of each other. So here's, same thing. So part C. So this time we're looking for a 1-to-1 ratio of beard to no beard with females. Yeah, I read that. Oh, I can read things backwards. I'm getting better, guys. So I did my cross. I'm trying to figure out how to get the females to have a 1-to-1 ratio, which means that I have to have big B's across and just 1 little b across the bottom because that's the dominant allele with females. So one parent would have to be homozygous dominant, well, in the way it looks, and the other would have to be heterozygous. So, the mother is heterozygous while the father is dominant homozygous dominant. There we go. So then part D, same question, but this time we're looking for the males and having that 1-to-1 ratio again. So it's kind of amusing because it's not the same at all. Unlike the first two parts up here, where it's more or less you just flip-flop. These ones down here with because the big B is what's dominant to get a beard. So the mother is heterozygous because she doesn't have a beard in the question. I don't think. Oh no, it's just saying here's the ratios, figure it out. So the mother is heterozygous while the father is homozygous recessive. And that's how you get a 1-to-1 ratio for males in this problem. So the last question is talking about X-linked inheritance and reciprocal crosses. So what I have done is the first part, so there's 4 parts again to question 5, in case I didn't say that already. And so the first part is saying, it says, in cats, orange coat color is determined by the B allele, and black coat color is determined by the B allele, in the sense of capital B. Sorry. The heterozygous condition produces a phenotype known as tortoiseshell, and big B and little b alleles are X-linked. Suppose you cross an orange cat, male cat, to a tortoiseshell female cat. What are the genotypes and phenotypes of the progeny? So to start off this question, I wrote everything down so that way I could remember. And then, so this way I wrote down that the male is orange, so that means it has to be that kind of cross. And the female is a tortoiseshell, so that means she's heterozygous. So then I went about doing my cross and got that you get a little bit of everything, sort of, in the sense of the female right here is going to be the tortoiseshell. I just kind of shorthanded or abbreviated. That's whatever. And then the bottom here is that the female will be orange because it's recessive. And the male will be black, and the other male will be orange because you got dominant recessive. So question B is part B is asking about question A, or part A. And in this one, it's asking about how do you get a male tortoiseshell cat. So my thought was maybe that in order to get what we need, there would be 2 X's. So there'd be 2 X's and a Y for this male cat in order to get the tortoiseshell coat. Now, I'm not quite sure if that's the case because I was thinking about how sometimes the X chromosomes go inactive, but I think that's with females. I don't know if males have the same. I'll have to go back and look. If they have the same makeup in the sense of like, well, you have 2 X chromosomes, one's going to be a bar body, so now it's inactive, or something like that. But so that was my thought process with the tortoiseshell male cat. So moving on, part C of question 5 is going into fruit flies. So the question is, a mutation produces a scalloped phenotype, which is an irregular margin on the wings. Assuming the scalloped phenotype is the result of an X-linked recessive mutation, indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. So I went about writing this all down as usual. So I have everything right here. So the scalloped and the narrow or normal wings and how they would look. The fact that, this is dominant, that's recessive. Here's the female's makeup. She's dominant, homozygous dominant, and then he's recessive. And so I did a cross between them to figure out all the progeny. And so you have all these guys. So you have the heterozygous females, and the males are both dominant for the normal wing. And so I just wrote that all of these children will be normal. Which is funny to me. Anyway, part D is also asking about the wings, but this time they say that in part C, assume the scalloped phenotype is the result of an autosomal recessive mutation. It's no longer dealing with X chromosomes at the moment. So indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. So once again, this time I don't have to, pardon me, I don't have to worry about this. So I just have them displayed with that, the female's homozygous dominant, male's homozygous recessive. I cross them. Everyone is the same. So you have heterozygous children, once again, with more normal wings. So that is it for everything. Um, may not have made a lot of sense. But if you have anything that may help me learn more, go ahead and put it in the comments, or I will be around. And thanks. See you next time.